Now that we have pretty much covered the understanding of the structures and functions of many different microorganisms and have a general sense of the kind or the variety of microorganisms that are existing, we will now consider how the human body and various microorganisms interact in terms of health and diseases. So as you can see, this chapter is actually going to be covering disease, epidemiology, and pathogenicity. So we will explore each of them. Firstly, what are diseases? Diseases are caused by a pathogenic microorganism. And when our defense resists these capabilities to fight that invasion, we get sick, and we're carrying a disease. After the disease has established in an infected person, some will suffer temporarily, or sometimes it may be a permanent damage, or in worst scenario it could be fatal conditions. So we will, this chart is very interesting because it shows the portal of entry for many of these pathogens, and we will go into these later too, but you can see the mucous membranes, whether that's the lining of the respiratory tract, the GI tract, the urinary tract, or the eyes, or the conjunctiva and the eyes, are the mucosal entry of of a microorganism. Another entry is through the skin and parenteral. Let's look at some definitions up here. The first one is pathology. Pathology is mainly the study of diseases. Pathos is a word for Greek origin, which means suffering, and logy is always study. So the study of mainly suffering, which would be pathology. Pathology is mainly concerned with the cause of the disease, and the cause of the disease is also called as the etiology of the disease. So what caused it? What were the different factors that would start this disease? Now, secondly, we deal with something called as pathogenesis, that is the manner in which the disease develops. The third part of pathology is concerned about the structure and the function that are caused by the disease and how it affects the human body. So sometimes we look at when a person gets sick, and we sometimes use the terms infection and disease simultaneously. However, in this particular study of pathology, there is a difference between the two. Infection is mainly by an effect of the invasion of a pathogenic organism. Remember, pathogens are your disease-causing organisms. So when there's an invasion, or certain colonies of these organisms develop within the tissues, that's called as an infection. Disease will follow infection, and that is the infection may result in a change of state from unhealthy to, for example, if you have a sore throat, then that is causing the infection of certain organisms such as S. aureus towards the throat, is then causing you the change of state, which is a sore throat or fever and so forth. Disease is pretty much an abnormal state in which the body is not properly adjusted to do certain normal function. And epidemiology is actually the study of all of this. This is, when did the disease take place, where did it take place, and how is it transmitted? So here are some epidemiological terms that you should know. Most of these are from a source that is the CDC, which is the Central of Disease Control, and that is your U.S. Primary source for all of these diseases. So, for example, when you work in a hospital or a doctor's environment, any kind of disease that takes place, which is slightly not as considered as a normal disease has to be reported to them, and they study how many cases of certain infections are taking place. For example, if there is a salmonella poisoning or a disease that is spread by a lot of people in an area are getting sick, then they will ask what were taken, how many people were getting sick and what were they ate. So they study all this whole study, which is called as disease control. On the table on the top, you can see there is a section called as emerging infectious diseases, which are all the new diseases that are coming up. And you can see up here, for example, you can look up in the book, this particular table 14.6. I couldn't get all of that very clarity on this slide. For example, the first one is listed as Bacillus anthrax. That's the organism, and the year of emergence of this organism to cause a disease is 2001. And what did they cause? The disease was called as anthrax. And then you can study more about it. For example, then another one that I want you to note is the one that is all the way down below, that's called as E. coli. It's been there since 1982, and E. coli will cause hemorrhagic diarrhea. So a lot of fluids are lost through this particular disease. So here's a list of bacterial diseases. Then you have a similar table, which is the next one, which is about the fungi. And then you have a list of protozoans. For example, notice trypanosoma, which was discovered in 2007, and that is a Chagas disease in the U.S. it's causing. Then you have helminthes, little creatures that cause raccoon roundworms or encephalitis in humans. And then you have a list of viruses that cause these different diseases and the year that emerged. And lastly, notice up here is the prions. Prions are your mad cow disease, and these were not really, prions are actually protein segments, and they will cause this disease, and it's been primarily reported in the Great Britain area. So coming back to the terms on the top, morbidity is the incidence of a noticeable disease. So, and mortality is how many people in that area have covered this disease. Sporadic means that it is happening throughout. Actually, I'm going to check on this recording for a second. A sporadic disease is a disease that it can occur occasionally, such as typhoid fever in the U.S. is such a disease. Then we have diseases that are called as endemic. Endemic diseases are those that will always be present in the community. For example, the common cold is constantly endemic in the community. Then we have epidemic diseases. Epidemic would be then a sudden development of a disease in a very short time. For example, the famous the Spanish flu, that would be an example of an influenza that happened and spread worldwide, spread all over, and when it's spreading all over, it is considered to be then pandemic, because it's not just localizing in this area of the U.S., it's already gone over. So an epidemic will suddenly become a pandemic situation. Here, this particular example is shown with the outbreak of SARS, which is which would be an epidemic, because it was happening in China, but they were afraid of happening it pandemically. Again, another one we want to look at is the trends in overweight in adults, and that's also, you can see that it is getting more and more people are getting obese, so that's another fright to look at. What kind of microorganisms do we normally carry? And those are called as the normal microbiota or the normal flora. The number for that is 1 in the ratio of 10 to the power 13th. So pretty much there are lots of organisms that we carry that are part of our normal condition. A typical human body will contain about 1 times 10 to the 13th number of body cells, and we contain about 1 times 10 to the power 14 number of bacterial cells. That is almost 10 times more bacterial cells than the human cells. This will give you an idea of how many organs we are harbor in our body. But we're not always sick. It's because a lot of them are present as normal microbiota. They are not disease causing. And when they change from a disease causing to a flora that will then it was considered to be transient, because they may be present for several days, weeks, or a month, and then they will disappear. Microorganisms are not always found throughout the entire human body, but they are definitely more abundant in certain regions. For example, up here in the nasal epithelium, you can see the orange, all these rods, and these are in the lining. Then you have a lot of them in the lining of the stomach, and then you have a bacteria that's lined up all over the large intestines. So these are just some of the very common kind of bacteria that are present in certain regions of the body. They can be introduced at time of birth and after. For example, lactobacillus can enter a birth canal, and E. coli can also change locations. And when actually it changes from the normal place where it's supposed to be and goes to a different region, that is when it will cause a disease. For example, E. coli is usually present in the large intestine, and they contain large number of this in the they are constantly being shed off through the lining of the intestines, and some of them sometimes are flushed out through the normal microbiota. But if they go in a different part of for example, it goes into the cervix region, it can cause you infection there. I'm going to go into more of these in the upcoming slides. So let's see. What do normal flora do for me? So microbial antagonism, as I just said, is a phenomenon in which organisms are competing among the microbes, and they once a normal biota is established, it can benefit the host by preventing the overgrowth of these harmful microorganisms. One consequence of this is that the normal microbiota protects the host against colonization by potential pathogenic microbes that are competing for the nutrients that are produced or the substances that are harmful to the invading microbes. And such conditions that, for example, a change in pH and available oxygen may lead to this competition. And when this balance of the normal microbiota and pathogenic microbes is upset, that's when the disease is going to take place. So, for example, E. coli releases a bacteriocins, which increases the formation of lactic acid and maintains low pH, and this is actually this low pH actually inhibits the overgrowth of the yeast, Candida albicans, which can grow when the normal microbiota is upset, and the pH is altered. So when you have a high pH, then there's a chance of yeast infection. We will talk about different relationships here. A flora can be symbiotic. Symbiotic means that the relationship between the normal microbiota and the host is such that two organisms are living in which one organism is dependent on the other. And this kind of relationship is called as commensalism. The example of this is Corynebacteria on the eye. that because that inhibits the surface of the eye and certain saprophytic microbacteria that will inhabit the ear and the external genitals. These bacteria actually live on secretions and slough off cells, and they bring no apparent benefit or harm to the host. So that's a commensalism. So symbiosis is pretty much a relationship between the two, the normal and the host cell. Another kind of relationship that we want to look at is mutualism. Mutualism is a type of symbiosis that benefits both organisms. For example, the E. coli in the large intestines will make a vitamin K and some B vitamins, and these vitamins are absorbed into the bloodstream and distributed by the body. In exchange, the large intestine provides nutrients for the bacteria and resulting in their survival. Another kind of mutualism is called as opportunistic pathogens, and these are microbes, for example E. coli. These do not cause disease in their normal inhabitat in a normal healthy person, but may do so in a different environment. For example, the microbe can gain access through a broken skin or a mucous membrane and cause opportunistic infections. Another kind of relationship is parasitism. Parasitism is when one organisms benefit by deriving nutrients at the expense of the other. This relationship is will cause many diseases because many bacteria can be parasites. How was the disease etiology determined? The disease etiology was studied by Koch, and Koch did his famous experiment that we mentioned earlier in the beginning chapters, in which we mentioned that he established certain postulates, and he has which determined that microorganisms cause disease. His famous case is the disease of the cattle that can also occur in humans, and he pretty much showed that certain bacteria today that are known as Bacillus anthrax were always present in the bloods of the animals that had the disease and not present in healthy animals. So, and pretty much from working on from this diagram, we can see that the microorganisms are isolated from a disease or a dead animal. The microorganisms are then grown in a pure culture and injected into a healthy animal. The microorganisms are also identified. The disease is reproduced in the second animal, and the microorganisms are again identified, and they're similar. So he was actually able to determine the etiology of the disease, that is the cause of the infection. Once you know what the disease is caused by, you can then develop certain antibiotics against that particular pathogen. So to understand the cause of the disease is very important for any kinds of vaccines that can be developed for the disease. Are there any exceptions to this study? Yes, there are some exceptions. Some microbes have unique cultural requirements. For example, the bacterium Trypanomema pallidum is known to cause syphilis, but the virulent strains have never been cultured on artificial media. And the causative agents of leprosy, that is Mycobacterium leprae, has also never been grown on artificial bacteria. So sometimes these are very difficult to grow in artificial conditions. The discovery of such microorganisms that cannot grow on artificial media has necessitated some modification to Koch's postulates, and the use of alternative methods of culturing and detecting certain microbes have been studied. Sometimes many bacteria, such as pneumonia, are also difficult to grow outside the conditions. On the other hand, there are bacteria that can cause several diseases, such as Streptococcus pyogenes, that can also cause scarlet fever, strep throat, or osteomyelitis. And the reason it does it in different conditions is actually based on the environment. In this section, we want to study how are infectious diseases classified. Every disease that affects the body somehow will alter the body's structures and functions, and the patient will show these signs through something called as symptoms. Symptoms are mainly changes in the body function, such as pain or a feeling of fatigue and tiredness. These are all signs that, and it's a pretty objective analysis, because some people have a stronger pain threshold, and it's very difficult to measure pain and these signs. However, a specific group of signs are called as syndromes, and these signs of a specific symptoms are actually easier to diagnose and associate with a disease. Any disease that spreads from one host to another directly or indirectly is called as communicable disease. For example, a direct is shown to you up here with this holding hands, or sneezing, or sharing supplies. Certain diseases such as chickenpox, measles, typhoid fever, tuberculosis, chickenpox are also examples of contagious disease. That is, the disease are easily spread from one person to another. On the other hand, a non-communicable disease is the one that is not spread from one host to another. These diseases are caused by microorganisms that normally inhabit the body and occasionally produce disease that will be that can be detected. For example, Clostridium tetani produces disease only when it is introduced into the body via abrasions or wounds or cuts. Okay, we want to know some more terms about the diseases, which will detect the severity or the duration of the disease. So here are some more terms. The first one is an acute disease. An acute disease is one that develops rapidly but lasts for a very short time. The best example is influenza would be considered as an acute disease. A chronic disease, on the other hand, is the one that will develop very slowly, and the body's reaction may be less severe, but the disease is likely to continue to occur for a longer period. For example, infections with mono or tuberculosis or hepatitis B fall into this kind of category. A disease that is intermediate between acute and chronic is called as the subacute disease. An example of subacute disease could be a sclerosing pedencephalitis, which is a very unusual brain disease that is characterized by diminished intellectual function or loss of nervous activity. Another term is a latent disease. A latent disease is one in which the causative agent remains inactive for a very long time, but then it becomes active to produce symptoms of the disease. And we discussed this earlier too, and that's the one that is a chickenpox that will cause shingles because it's been residing in the nerve cells through the skin for a very long time. Nosocomial infections are those infections that are caused due to a hospital setting. So when patients are present in a hospital, or people who work in a hospital, the transfer of those diseases are called as the nosocomial infection. Over here we again have a lot more terms. And how else can infections be classified? That is the extent of host involvement. The first kind of involvement we want to classify is the host body, which is affected, and that is called as a local infection. A local infection is one in which the invading organisms are relative to a certain area of the body. For example, you could have an infection in the eye, or there's here's the image that's shown over here. In localized infection is an infection under the armpits. So it's in general one area. Now, sometimes the local infection is always better to be taken care of at that particular region, because you don't want this local infection to get into a systemic infection. A systemic infection is when the organisms will now enter the bloodstream or lymph, and it will spread throughout the body through the circulation. Very often, these infections that enter a body or a lymphatic vessel will reach the other part of the body, and they will cause an infection spread, and that is called as a focal infection. That is, for example, that you can get an infection in areas such as your teeth, tonsil, or sinuses, because you had some other infection that was not taken care of. Now, once the primary infection becomes acute and it is not taken care of, which is the first part of the illness, a primary infection then can lead to a secondary infection. And those of you who have children, you must have heard that a lot when you take them to the pediatrician. Often it could be just a common cold, which is caused by a virus, but the physicians are always concerned about a secondary infection that will be caused by an opportunistic pathogen after the primary infection has weakened the child's or the person's body's defenses. Secondary infections of the skin and, as I said, the respiratory area are very, very common, and sometimes can be dangerous because they could lead to a more drastic state. For example, a cold can eventually turn into a pneumonia kind of case. Then those are called as the secondary infection. And the example over here is the pneumocystis, which is a pneumonia, which is a consequence of AIDS, is an example of a secondary infection. Bronchitis, which follow influenza, is an example of a secondary infection. Subclinical infections are inapparent infections that are not causing disease. For example, hepatitis A virus, or something like a polio virus that can be there, but it's not really causing any noticeable disease. All of this can be then be affected by predisposing factors. Okay, what are the 3 stages of disease development? And the graph up here speaks a lot, and you have the first stage, which is called as the incubation period. The incubation period is the interval between the initial infection and the first appearance of any signs or symptoms. Many times the incubation period can be a short time, for like a day or two. It could be 14 days. And you're pretty contagious during that time, but you're carrying the disease, not showing, you're carrying the infection, not showing signs of the disease. And that can lead to a prodromal period, that is a relatively short period in which the incubation is now going to cause disease, for example, general aches or tiredness and so forth. Now then it follows the period of illness, which you can see up here is the most, it's peaking up with the bell graph, and you can see the disease is getting more and more severe. The person will give signs of symptoms of the disease. You'll have more muscle ache, fevers, chills. Some people may get sore throat, or some region of the body, for example, lymph nodes can get enlarged. These are all period of illness. So that's like all the signs and symptoms when you go to a physician and they're asking you. Generally, the patient's immune responses and defenses have been overcome by the pathogen, and that's why you start getting sick. Now, this periods of disease, of illness, is then followed by periods of decline. That is, the signs and symptoms slowly start getting away, and you start feeling a little better. This can last up to 24 hours to 46 to several days. Again, this gives you a real interesting factor. Do you really need to get an infection, an antibiotic, or it will just go away with its time? Some people think that it will, but again, I'm not going to convince you on that, because that can lead to other focal infections as well. And then you have the period of convalescence, that is, a person will again get strength, and this will be a recovery stage. Here's a nice picture which shows how diseases can spread, and you can see the diseases can spread in something called as a contact transmission or indirect transmission or droplet transmission and so forth. So pretty much we're looking at the transmission as how the disease is going to be taken from one person to another. In any kind of pathogen, and you know, the infection or the pathogen is going to need a reservoir, and the reservoir could be humans, animals, or non-living. So pretty much we can get sick from each other, from animals that are carrying the disease. For example, up here you can see the fly that's been sitting on maybe some unhealthy environment and then comes and sticks on your burger, and if you eat that, here's this water and soil that can leads to infection. For example, the spread of cholera and anthrax or Clostridium is spread through these non-living factors. And then you have the human that are like, you know, they haven't washed their hands clearly. You have direct contact, sort of like touching and kissing are reasons of direct contact. Sometimes you could be using a utensil and you haven't cleaned up, you don't have your clean hands, you go and use a sharp knife, you cut some veggies or something, you put it in the food, and that's another way of carrying these infections. Yeah, pretty yucky, right? So here again, diseases are transmitted through direct, and you can see the contact, kissing, contact. Here's a vertical that is from a mother to a child. A vector would be something like a fly that is being used, or a mechanical or a biological vector. So pretty much I gave you some of these examples previously, but we can see this from different angles. Vectors are pretty much arthropods that are most important groups of disease carrying, and these could be, for example, your mosquitoes or your flies and all these little creatures that fly around. Those are all vectors. Sometimes birds can be vectors too. Houseflies are the most common vectors. Biological transmission could be, for example, a fly comes and bites an infected person and injects some of its blood, takes it to a different person, and now you've ingested it over there. So that's like a biological transmission. Direct and fomite, again, as a non-living object that has helped spread disease, and those would be water and soil or just brooming. And here's a list of indirect ways of transmitting diseases. Okay, what about nosocomial infection? Nosocomial infections are infections that are received during in a hospital stay. So pretty much it's really a leading cause in the fourth leading cause of death in the US, and it's increased in the last 30 years. Nosocomial is pretty much hospital acquired infection. That is people who work in the hospital, or even patients who have low immunity, who have less I'm sorry, I had a little program that came up. So coming back to the nosocomial infection. So you can see up here the urinary tract infection is the most common. Then you have the surgical sites, and then you have respiratory and other GI, and then you have skin and septicemia. So these are all examples of nosocomial infections. Nosocomial infections can be controlled, and the most important for that is hand washing, clean respirators, humidifiers, bathtubs, using enough alcohol wipes to get your disposable these objects clean, and disposable tools and bandages. So pretty much all I have all those features that as a nurse you will be, or as in a medical community you will be asked to take care of. Accredited hospitals will usually try to control these infections. Many infection hospitals have at least infection control nurse or epidemiologists who are actually there to identify problems causing such resistance strains of bacteria and so forth. For example, the MRSA scare is also because you've got overdose of bacteria of antibiotic, but again also a lot of more people are also transmitting it through not proper the proper techniques.